So we're looking today at how scientists figured out that DNA is the genetic material. They thought it was probably proteins, but they really weren't sure whether it was DNA or proteins. So they set up some experiments to figure this out. So we're going to start with Fred Griffith. He was actually not trying to figure out the genetic material. He was actually trying to figure out how to make a vaccine against Streptococcus pneumonia, which is a bacterium that causes pneumonia. And he didn't end up making the vaccine, but he found out some really interesting stuff on the road to figuring out what the genetic material was. So he isolated two strains of this bacterium, and they looked different under a microscope. This one was all bumpy, and this one was smooth. So he called this one rough, and he called that one smooth. So one strain had a rough surface, and he called that the R strain. And the other strain had a smooth surface, and that was the S strain. So the rough strain is harmless. You can get that and live. And the smooth strain looks smooth because a bacterium produces a polysaccharide capsule around themselves. So these two strains are very similar. This one has bumps on it. These bumps are antigens. And do you know what the body does with antigens? The body makes antibodies against it. And so your antibodies will come along here and attach these things together. And then your white blood cells will see, oh, look at all these antibodies here. This must be something bad that I need to attack. And your white blood cells will come and eat these up, and they'll be gone, and you'll win. Unfortunately, these have those bumps, but they're covered up by this capsule. And so polysaccharides really, they're not good antigens alone because they don't have any easily identifiable markers on them. They're all kind of the same. There's nothing special about them like one protein compared to another. So when these have the polysaccharide coat on them, there's no antibody to attach. You don't make antibodies because there's no antigen really to attach to. So these bacteria grow and grow and grow. They reproduce. Remember, by binary fission, and your body really can't do much to protect itself. So our immune systems have a hard time recognizing these. So this strain can kill you. Bad. The words virulent or pathogenic are used to describe them. Both of those words mean disease causing. So you can say, how virulent is this disease, meaning how likely are you to die? Or pathogenic also means, something that can hurt you. Here's Griffith's famous experiment. He injected the different strains into different mice. So this is a little bit sad for those of you who might be sad if mice die. So be forewarned. So he injected the live R strain. These are the non-pathogenic ones into the mice. Remember, this is the kind that your body can identify and make antibodies against. Once you have these antibodies, then your body can come along, your white blood cells come along and kill it. So you kill the bacterium before it kills you. Same thing for mice. So the result is that the mice survived. Yay, happy mouse. So there's the mouse before and after, and it's still alive. So then he would take a little bit of the blood from this one on the survivor and see what the blood looked like. And he found out that no R strains were isolated from the blood. So the immune system of the mouse must have been able to fight off these bacteria. Then he took this bad strain, the S strain, the pathogenic one, and he injected that into the mouse. And guess what happened? You got it. This thing reproduced and reproduced and reproduced, and it killed the mice. So the mice died. Sad. So the live S strains were isolated from the blood. So he took this dead one's blood and looked at it under a microscope, and he could see the S strains there. So the mouse's immune system might not have been able to fight these off. And then here's another kind of crazy part. This is understandable. He heated up, or predictable, I guess. He heated up this S strain until it killed the S strain bacteria. So he took some of this stuff, put it in a beaker, put a flame under it, heated these bacteria up until they were all dead, cooled it down, injected that into the mice. So he injected the heat kills S strains into the mice. So in other words, he injected dead pneumonia bacteria into the mice. and the mice lived. And then he looked at the blood of these mice and could not find any bacteria. So it turns out that if you kill the bacteria, um, understandably enough, the mouse lives. You can get dead bacteria injected into you. So this kind of makes sense because he was trying to make a vaccine. So that's what he was working on. Here's the crazy part. You ready? Crazy, 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 crazy part. He took these S strain bacteria and he killed them. Dead bacteria. Whoops. La. So he took the heat-killed S strain bacteria and mixed them with live R strain bacteria. So these are dead. I don't know why my pen's doing that. These are live, and he mixed them together. Mix, 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 mix. So these are dead. The bad ones are dead, and the good ones are alive. Should be no problem, huh? Wouldn't you think? No problem. And he took this mixture of the heat-killed S strain, the bad, dead ones, and the live R strain, meaning the live, good strain, and he injected them into the mice. And guess what happened? The mice died. Isn't that crazy? And guess what he found when he checked their blood? He took their blood out, looked at it under a microscope, and found the bad ones. All these bad ones, remember, had been killed. It was only the good ones that were alive. These are the S strains, right? They're dead, the bad ones. And then he injected them into the mice, and as it turned out, the mice died. And what he isolated was a bunch of these, the ones he had killed. Crazy, crazy, crazy stuff. So here's a summary of it. He injected the good strain, the R strain, into the mice, and they survived. Injected the live S strain, the bad one, into the mice, and they died. Injected the heat-killed S into the mice, and they lived. Injected the live good ones and the heat-killed bad ones, and they died. And then he found live S strains isolated in the blood. So he had killed the S, but then they found the S. Crazy. So the heat killed the S strain bacteria, but did not destroy the DNA. Did not destroy what he didn't know it was DNA, but did not destroy the genetic material, including the part that specified how to cause infection. So somehow that material had been transferred from the dead S strain to the live R strain. So these cells, the live ones, were able to actually pick up the DNA of the dead ones. That's called transformation, and it's something that bacteria actually do. So you can have bits of DNA that are just floating around, and you can have bacteria that are live bacteria, and they can actually take up the DNA and change themselves, change from that rough form to knowing how to make a capsule because they have the DNA from the dead one that said how to make the capsule. So that's called transformation, and it's something that bacteria do. So further experiments showed that the harmless cells, the R ones, had picked up the pathogenic genetic material, or DNA, permanently. They were able to cause infections for hundreds of generations. So he would have them in a petri dish. Here are the S strain ones. And take them and put them in another petri dish and grow up more colonies and take them and put them in another petri dish. And hundreds of generations later, they could still cause infection. So the ability of bacteria to take up genetic material from the environment was called transformation. The genetic material was called the transforming principle because they still didn't know if it was DNA or proteins or anything else. And I think I'm going to stop with Griffin's experiments